Stem cells are big news these days, not just in the American presidential election, but in science, and there's good reason for that. They're incredibly versatile cells. They can be turned into many different kinds of cells, and so there's big hope that they can be used to treat damaged cells and tissues, whether by injury or disease. There's new hope especially for being able to restore vision when eyes have been damaged or diseased, and that's thanks to a new breakthrough experiment that's just been published by researchers at the University of Toronto. And Brenda Coles is one of those researchers, and I'm with her in her lab right now. Hi.
Hello.
So what is the big breakthrough that you've done here?
So what we've discovered is that there are some stem cells that are found in the human eye, and we've shown that if we take those cells out, that they can proliferate from one single cell to about 10,000 cells in a dish. And if we put them on a sticky substrate, they make all the cell types of the eye.
Wow. Now, before we go any further, I know you have this model of an eye for a good reason. So let's show us where these stem cells come from.
If we open up the eye, this being the iris, look right on the inside. There's this little ruffled membrane here, and it's a pigmented cell, and one out of 500 of those cells are actually a stem cell.
Even adults have these cells with this potential to become other cells.
Yeah, and we're actually born with the same number of stem cells that we have by the time we're 80 years old.
Now, you've been able to take these cells, grow them up, and show that they change into other types of, how did you do that, into other cells of the eye?
So when we did it in a dish, it was just the growth factors, or like the nutrients that they were in, that actually just, they randomly turned into all these cells. Right. But we actually went a step further, and we did the transplants of these human retinal stem cells into a mouse eye, and showed that they can actually integrate and form the photoreceptors and the RPE cells, which are the cells that are lost in macular degeneration and retinal pigmentosa.
And they do that in the mouse eye?
Yeah, which is really exciting.
Can you tell the difference? If you look at a mouse eye, you can see which cells were human cells that were transplanted into it?
Yeah, so we actually fluorescently tagged our cells so that they would fluoresce green under our microscope if they were human cells. So when we look at sections of the eye that we've done these transplants in, we can see these green cells that have integrated, and the eye actually looks perfectly normal. There's no damage that's actually done from these cells.
So what's the significance of having done that much, taken it that far, Brenda? You can show, you can take these human stem cells and they seem to do the right things in a mouse eye.
What this actually shows us is that our cells that are quiet for some reason in our eyes actually have the capability of actually possibly regenerating our lost cells damaged either through physical or genetic diseases.
Now, where do you have to go from here? I mean, what's the next step along that road?
Yeah, so we're going to move back into a mouse model and take the mouse stem cells and transplant them into retinal degeneration mice, and show that whether these cells can first survive in that environment and whether they can actually functionally recover their vision.
Because you haven't actually, with the experiment so far, you haven't actually shown that these human cells are working in the mouse.
No.
Eye. They're just there.
No,
they've done the right things, but you can't tell whether they're working, I guess.
No, it's very hard to actually tell in a healthy animal whether our cells are actually making the right connections and signaling to the brain. So we'll actually have to eventually do all these experiments in a blind model and show that we actually get vision.
Right, so you'll be taking mouse stem cells, putting them into mice that are blind?
Yes.
And then trying to see if it makes them able to see again, I guess.
Yeah, so we'll be doing that through behavioral studies or through ERG, so electroretinogram, just measures whether there's signals happening.
It seems really complicated to me, because there are you've got to get connections from these light-sensitive cells actually to nerves, and then to the optic nerve, and then to the back of the brain. So there's a lot of connecting to be done there.
Yeah, it's a big step that has to be taken. But the fact that these cells, when we put them into the environment, are actually connecting and going to the right place, gives us hope that maybe when we actually put these immature cells in there, that we can either give them signals that will actually help them go to the right place and make the right cells, or we'll have to make sure that we do it in an earlier time point.
Well, it sounds like lots of exciting experiments to be done, but this one's pretty good too. Thanks, Brenda.
Thank you.
Brenda Coles is a neurobiologist here at the University of Toronto.